Let's see, I guess I'm going to start off by introducing my co-authors to you all. Most of you probably won't know these co-authors because they are all fish ecologists. So they do some really sophisticated population ecology, and I told Dr. Forsyth I wanted to do something similar with spiders, and this paper is one of the results of that. So introduce you to our focal species of this project. This is Drapetisca alteranda, a medium-sized sheet web spider, which is a tree trunk specialist. When it becomes adult, it leaves the leaf litter and hangs out in the lower tree trunk of mostly deciduous trees in the late summer and autumn, and it makes a very flimsy little web, and it mates on the tree trunk, and then the females lay their eggs back in the leaf litter, and the cycle continues. Very closely related, very morphologically similar species in Europe, Drapetisca socialis, has been studied pretty extensively in the 1990s by Karen Schutt, and she documented that the spider does not use its web to forage with. Instead, it has a cetal trap made of huge macroceti on its pedipalps and its chelicerae. And so here you see a normal Linyphiid with sort of its normal ceti, and then the giant macroceti of Drapetisca socialis. So Drapetisca alteranda, the North American species, is very similar morphologically, and we believe it does the same thing. Here's our study site, deciduous woods in northeastern Wisconsin on the campus of UW Green Bay. And it is a 1 hectare forest dynamics plot. And you can see it's made of 24 20 by 20 meter subplots. And it's on a floodplain of a little creek, Mahone Creek. And you'll notice that there is a steep slope in the middle of the plot. And so there's kind of floodplain at the bottom and uplands at the top. And here's an aerial photo of the site in these, what we call Mahone Woods. Mahone Creek flows down to the Bay of Green Bay, the largest freshwater estuary in the world. And here's UW Green Bay over here. This is where my office is located right there, so very convenient. Now, really importantly to this study, this forest dynamics plot project, every single tree diameter breast height greater than 10 centimeters has been mapped, measured, and identified by botanists. So this is a map of all the trees. It's over 1,000 trees on this 1 hectare plot, and we have sampled them all for this particular species. So this is what the photo of what the site looks like in the autumn. It's obviously a really nice place to do field work. So this is a great example of sort of integrating teaching and research. So I need to thank a whole bunch of students in this environmental science capstone course that helped us with this. And actually, the first time we did this was in the fall of 2012, and we divided 14 students into different teams, and they worked on different aspects of Drapetisca biology. And they would often randomly sample some of these 20 by 20 subplots to do their work. And that resulted in a talk I gave at 2013 AAS in East Tennessee, and then a note in Journal of Arachnology in 2014. So in the fall of 2014, we decided that an inventory would be way better than a sample. So if we've got a pretty big class, 18 students, and we thought if we all worked together, we could sample every single tree in this 1 hectare plot and get really nice data that way. And so that's the result of this talk. And then we also published a paper that came out last year in environmental entomology. The methods are really simple. We did our work in September 2014, and we sampled every tree in this 1 hectare plot. And of course, the trees were measured 2 years before, and all of that data is already there. What we did to find the spiders is brush with a horsehair brush onto a 1 by 1 meter nylon beading sheet. We brushed from the ground surface to 1.3 meters above the ground surface. There were these dendrometers on a lot of the trees at 1.5 meters, and we wanted to make sure we didn't disturb those. The previous work has shown that spiders are pretty rare above 1.5 meters on the tree trunk. So we got most of the spiders by doing this. When the spider was observed, its estimated position on the trunk is marked with a thumbtack, and then we could measure the height above the ground, compass direction. And we also quantify the depth of the bark using a dowel at 8 compass points around that estimated position. And then we captured almost all the spiders, all but one or two, and returned them to the lab. In the lab, the spiders were dispatched in the freezer. I confirmed the identity and sex of all the specimens, and it's pretty easy spider to identify. Students measured the carapace width of the spiders as an index of size. Two different students measured it independently, and if the results weren't close, then I would measure them again. And then each spider was dried at 60 degrees Celsius and weighed. So we, the royal we, sampled over 1,000 trees, 26 tree species, and we found 399 adult Drapetisca alteranda on 193 trees. That's 18% of the trees. Our sample was 62% female spiders. And we think this is because we kind of missed the mating peak, right? When we think there's basically 50%, but as we sampled, the proportion of males continued to decrease over time. So I think we missed the peak. Males die faster than the females, and so we have a female bias sample. So just quickly looking at the spider and microhabitat variables, the main question is there a relationship between spider variables and the microhabitat variables? And so we did all sorts of ANOVAs, t-tests, circular ANOVA for the compass direction. And we used Bayes factor to assess whether these differences are negligible or positive or strong. And the really short and easy story is that none of the spider variables were significantly related to microhabitat preferences. The Bayes factors were always less than 1.7, which is a really low number. They can go up to 150 and more. We did find some interesting things with the spider data, though. I've sort of reconfirmed that in this species, sort of rarely among spiders, the males are bigger than the females. They almost always have a carapace width bigger than 1 meter, and the females are usually smaller than 1 meter. And then you can see, though, that as usual, females outweigh the males. So females almost always weigh more than 2 milligrams, and males almost always weigh less than 2 milligrams. This is just showing the vertical distribution of a bunch of the tree species where the spiders were found. And you can see there's not a big difference between tree species. And so the main thing I wanted to show you here is just the average height at which we found spiders, which is about 83 centimeters above the ground surface. They don't go very high. One thing that definitely is important in predicting spider abundance is the tree species and the size of the tree. So all Drapetisca prefers larger trees, especially smooth bark species. So what we're seeing here is modeled posterior predicted abundances on different tree species. Each line is a different tree species. And as the tree gets bigger, you always get a few more spiders on that tree. But some tree species, the spiders really prefer them. And so the slope is much more positive. So this is paper, birch, and quaking aspen. So those are some of the smooth barked species that these spiders really prefer. So now I want to talk about this model that we wanted to do to model the spatial distribution on this plot and try to recover the empirical data that we got. And so what I'm showing here really is just the empirical data. It's a heat map, but basically this is the raw data of where we found spiders on the 1 hectare plot. And we attempted to model the probability of spider presence on each tree in the plot to make a modeled heat map. And we had random effects from previous analyses that we thought were important tree species, and then also spatial autocorrelation, which I'll talk about in a second. And then our fixed effects were tree size and elevation. And some models we used both of them or neither of them. And so with all of our models, there are really two parts of this model. There's one model that describes the abundance in a tree when there are spiders there. And then there's another model that describes among all the trees whether there will be a spider on that tree or not. And for that model, we used a zero inflated Poisson general linear model to compensate for this data that has a whole bunch of zero values. So basically we constructed 16 different combinations of pairs of models with different fixed effects or no fixed effects. And then we used Watanabe-Akaike criterion to sort of select the best one.
10 minutes have elapsed.
Okay, I want to talk about the autocorrelation real quick. Obviously, the presence of conspecifics is very often a strong predictor of the probability of species observation. So spatial autocorrelation is really kind of a proxy for who knows how many often unknown or unmeasured environmental predictors of species presence. So we think it's going to be pretty important, and we want to try to add spatial autocorrelation in this model. And you do that using a conditional autoregressive random effect. We just call it a CAR because it's really hard to pronounce. This is pretty computationally intensive. It requires an analysis of a neighborhood of trees around each tree on the plot. And so you have to select a neighborhood size. And so we tried different neighborhood sizes from 10-meter diameter to 40 meters diameter. And we used the smallest one that gave us stable results. So that turned out to be 20 meters neighborhood. These models were run 6,000 steps. We discarded the first 3,000, and then we looked to see if they would converge, and most of our models ended up converging. So here's the results. These are all heat maps of different models. Again, this is really the raw abundance data. This is our empirical data that we're trying to match. Map B here is really just the spatial autocorrelation without any environmental variables. And it actually reconstructs the distribution reasonably well, but it definitely underpredicts the spider abundance. Model C here is the best fitting environmental model without the spatial autocorrelation. So it, I would say, significantly underpredicts spider abundance, although it does look something like our empirical data. And then finally, we have what we think is the best model, which is the best fitting model with the spatial autocorrelation. And it seems to be a better predictor of the spider abundance. So basic conclusion of this is that the landscape scale, these meter scale spatial autocorrelation can actually be important in predicting spider distribution. So lastly, I wanted to look at factors that accounted for the distribution that we see on this plot. First of all, of course, you see that a lot of the spiders are caught in this sort of northwest corner of the plot. And this is where large individuals of Drapetisca's favorite trees grew. So there's a bunch of big green ash and American basswood, and those species prefer well-drained soils on north and east facing slopes. So that's where they were in our plot. The reason why there's not very many spiders down at the southern end of the plot is because of elevation, which is weird because there's only a few meters of elevation difference. So really what we're talking about is elevation is a proxy for topographical position. So in my experience, you can't catch Drapetisca on floodplains and I think it's because in the springtime, the flooding causes a lot of juvenile mortality. So that's what we think is going on here. So I just want to acknowledge First Nations people who are the original stewards of this land. I want to thank the people that set up and measured all those trees in the forest dynamics plot. And I want to thank about 40 UW Green Bay students who helped us in various classes with different aspects of this work. Thanks.
Thank you. So Mike, you got one question from Linyphiidae who asked, is it possible that your sampling was biased for smooth-barked tree species? Smooth-barked tree species.
Yes, I think there's no way to get around that. I think it's possible that that we can catch them easier on smooth-barked species, and I'm still trying to work out how would I separate. Find a different sampling method or something, but yeah, that's definitely a possibility.
I noticed that. What's the position of this plot again from distance from the water?
It's probably about 25 meters from the stream side. It's only about a couple inches in elevation. So
yeah, okay.
Well, it definitely floods. The bottom of it definitely floods in the springtime.
Yeah, but it's just more extreme in that one corner.
Yeah, and certainly water doesn't come up in most of the plot. It's just at the very low end. You get a few centimeters of water sometimes.
Bruce Cutler noted that there's the same male and female size relationship in Cinemosina americana, but not in most other Cinemosina. So that was a comment.
Oh, that's interesting. I was going to say that a lot of Linyphiidae turn out to have larger males than females. It's not really generally appreciated.
So more based on reproductive, or weight is more of a reproductive readiness.
Yes,
thing,
absolutely.
Differentiating males and females. So no canopy spiders. So I've always wondered this, and I'm not a spider person so much, although I'm getting there.
Honorary spider person.
Yeah. So are there spider canopy specialists, or are the same spiders that you might find in canopy found on the trunks, and so it doesn't matter. You can just sample the trunk and say you've gotten it.
It's definitely, there's not a lot of them in the temperate zone. There's a few of them that have been identified. For example, in England, there's a Linyphiidae that's been identified. As you get into the tropics, though, there's definitely some species that you almost always find in canopies, and they don't like lower down. But yeah. Okay. I think in temperate zone, it's pretty harsh environment up there, and there's not too many specialists.
Many canopy Linyphiidae. Oh, okay. It sounds like, I think we have to, or rather we get